Good morning, everyone. Welcome to Grand Rounds. My name is Alex Harris, and I'm one of the Grand Rounds co-directors, along with Drs. Adi Talati and Rebecca Mueller. Next week's Grand Rounds will be given by Dr. Deborah Hassan, Professor of Clinical Epidemiology here at Columbia University. The title of her talk is U.S. Cannabis Use and Cannabis Use Disorder in a Changing Cannabis Landscape. I have an announcement from the Continuing Ed Department, who asks everyone to please remember to fill out the CME survey. They're seeing about a quarter of the people who show up actually filling out the CME survey. If you'd like CME credit, fill out the survey. As a reminder, if you're participating via Zoom, you may post questions at any time during the talk. Please use the Q&A feature, not the chat feature. As the online moderator, Dr. Talati will forward questions to me for our speaker to answer. We encourage trainees to consider asking a question, and as you know the drill by now, trainees go first. Please indicate in the Q&A if you're a trainee. It's now my great pleasure to introduce today's speaker, Dr. Keri Martinowich, who's a senior investigator and director, not of the entire Lieber Institute, but of the Translational Neuroscience. And she's also professor in the departments of Psychiatry and Neuroscience at Johns Hopkins University School of Medicine. She earned her BA in International Relations from the George Washington University before pursuing a PhD in Neuroscience at UCLA, where she did her thesis work under the mentorship of Dr. Yves Sun, doing pioneering work identifying BDNF as the first brain target of the signaling molecule MECP2. She then did a postdoc with Drs. Bai Lu and Hosami Manji, studying neurotrophin signaling in circuits controlling behavior. Dr. Martinowich joined the faculty at Johns Hopkins University and Lieber Institute for Brain Development, where she leads a research team employing a cross-species approach to study how gene expression in specific cell populations contribute to circuit function and neuropsychiatric disorders. Her lab integrates genetic manipulation, viral techniques, molecular and cellular and systems level approaches in animal models with cell circuit and specific transcriptomic studies in the human brain, and human iPSC-derived culture models investigating how gene expression programs in cell type specific populations regulate circuit function and behaviors relevant to psychiatric disorders. Her work has been supported by numerous NIH and private foundation grants. She's a fellow of the American College of Neuropsychopharmacology and was recently honored with the ACNP Daniel H. Efron Research Award. You may also know her alter ego on Twitter, where for several years she was the witty and science trivia loving voice of the journal Neuropsychopharmacology, stirring up controversy on such weighty matters as the proper coloring scheme for neurotransmitters. The title of her talk today is Cell Type Specific and Spatially Resolved Approaches for Understanding Human Brain Disorders. Please join me in giving her a warm welcome.
Okay, can everybody hear me? Okay, I want to thank Alex. It's really fun to be here. I know so many people. I feel like I used to come here as a postdoc when my husband was working with Renee's lab, and so I used to come up here. So I feel like I've known people here for years, so it's just really fun to be here. So like Alex said, I'm going to talk today about a lot of our postmortem human work that we're doing in the brain to kind of isolate spatially resolved profiles and also cell type specific profiles and apply this to understanding complex brain disorders. I want to start, let me put this on. I just want to start by kind of acknowledging some of the people I work with. So we run for about these are big kind of omics approaches, take a village. And so a lot of these are in kind of collaboration with 3 junior faculty members who are in my division, Kristen Maynard, Leo Collado Torres, and Stephanie Page. Kristen does a lot of the postmortem human brain molecular profiling. Leo does a lot of our data science and also kind of making all of our stuff public. He's really awesome at this, making all the apps that hopefully a lot of people have used, our resources. Stephanie Page does a lot of the microscopy and imaging, which is critical for our spatial transcriptomics approach. And then Stephanie Hicks is an associate professor in the biostats department at Hopkins. And she's worked with us on developing a lot of new computational tools for analyzing these emerging single cell and spatial technologies. Okay, so I was told that I had to put some learning objectives on here. I don't know if these are good learning objectives, but I'll go through them. So basically, hopefully after I talk to you today, you guys can tell yourselves or your friends what omics technologies might be able to tell us about brain function or what they're useful for in both the normal and the disease state. Also how we can use omics technologies to help us translate rapidly kind of coming online genetic insights from GWAS and other types of functional genomics kind of information. And then also when using these omics technologies, why spatial resolution is important. So we've moved from cell type into spatial and like why that's really important and kind of critical for us to add in. I'm going to go first through sort of what we do at the Lieber Institute. For those of you who are not familiar with our institute, we were founded around 2011. I started at the Lieber Institute in 2012. So our institute was really founded to basically translate genetic insights that were coming online from rapidly kind of collecting GWAS information for neuropsychiatric disorders and think about how do you actually use that information and actually translate it into treatments, because we were getting these lists, which was awesome. Like we finally had GWAS hits, and then people were like, what are you going to do with that stuff? And so the approach that the Institute really took was to kind of capitalize on biological resources. So we have a very large human postmortem brain bank that now has, I think this is outdated, over 5,000 postmortem brains that we've collected. And then we also collect kind of paired dural fibroblasts from those donors so that we can go back into cellular models from those same patients that we're finding sort of molecular associations in the brain, and then take those back into cellular models to do causality and validation studies. We integrate this from both of these things with genomics and transcriptomics, and that's kind of the piece I'll talk a lot about today. And then kind of try and take this to understanding mechanisms of illness, and then go back into animal models to understand circuit function and neuronal cell models that we make from the IPS lines from these dural fibroblasts to really understand causality. And that's funneled into sort of medicinal chemistry and drug discovery to really try and take these things into novel therapeutics. So the approach that we've taken in my group, or kind of our division, is to really integrate information from human brain and model systems. And so those model systems being the rodent, and then also the human cellular models that we're driving, the induced pluripotent stem cell models. So really what we try and do is first start with the human brain, because we're really trying to understand human disorders. And so what we're working with the postmortem human tissue to identify cell types and then molecular signatures within those cell types that are associated with disease. And I'll talk today a bit about how we're kind of that approach of what we're doing and trying to get that information. And then I won't talk as much today about what we do with that information, but just sort of have a holistic view of what we're doing in the group is that we then integrate that with rodent data, that rodent molecular data that has experience dependent and circuit plasticity. And so the reason this is important, postmortem human brain tissue is a really valuable resource, but it has a lot of limitations. And so some of those critical limitations are that we can study brain regions, and we can study brain regions that are across circuits, but we can't actually study circuit function in a postmortem human brain. So we can't do all the kind of cool things that we can do now in rodent models, where we can actually like label projection neurons, or we manipulate projection neurons and then sequence in the downstream areas. So we can't get that actual circuit specificity. Also, it's postmortem human tissue, so we can't get behavioral experience. So people die and then we have the brains, but we're not doing experiments on people, so we can't have sort of pharmacological manipulations or some type of a behavior where we're sequencing afterwards. So our approach has really been to kind of capitalize on these two things, the postmortem human brain, where we have disease status and it's an actual human brain, and then integrate that with the rodent molecular data where we do have this, can get experience or pharmacology sort of exposure, and then also the circuit specificity. And from integrating that information, then we can prioritize those molecular associations of disease and kind of validate them down, prioritize them, which ones to investigate at cellular, behavioral, and network levels. We're really interested in a couple of defined circuits and specific behaviors, but we are generating a lot of kind of large scale consortium scale data across the human brain for other areas. And so we're also really passionate about making sure that that data from this valuable resource gets out there to the community. So a really kind of importance to us is to build biologist-friendly tools for interrogating the human data so that some of those brain regions that we're profiling, but we're not going to take back and do this kind of prioritize and really drill down on those molecular associations so that people can capitalize on this similar approach and kind of find these molecular associations in brain regions and diseases that they're interested in. Okay, so I'm just going to go through for you quickly sort of what techniques we do. I think most people now at this point are pretty familiar with some of the single nucleus RNA sequencing or single cell techniques, and then increasingly so with the spatial gene expression. So I think it's, I don't know, maybe about a decade now that single cell sequencing came on the scene. In postmortem tissue, we're really doing a lot of single nucleus RNA sequencing for technical sort of logistical reasons. But basically, these are kind of droplet-based technologies where we're sequencing. It just allows us to get molecular profiles from individual cells. These are really great technologies because we can actually get, profiles from single cells, but again, there's some limitations. That tissue just basically that we're taking from the human brain gets smushed up, and so we get single cells, but we can't put them back and know where they came from in place, which for people who study the brain, structure function is important. So we really want to know where they came from. The other limitation that I talked about is that just because of some logistical issues with frozen tissue, we do have to do single nucleus RNA sequencing. And so while there's really good correlation between single cell and single nucleus RNA sequencing that people have characterized, we are losing, the cytosolic transcripts, which is a problem. And a lot of the spatial gene expression assays retain those cytosolic transcripts. So that's important for us. And so I think especially as neuroscientists, while there is this really good correlation between molecular profiles in a nucleus and then just the overall cell, a lot of things that are out at the dendrites or in the processes, these cytosolic transcripts are a lot of things that we really care about, right? So those are a lot of things that contribute to genetic risk or things that we know are important for brain function. So losing them, for other types of science might be less important than those of us who study the brain. So with spatial gene expression, basically you can get this, that you can actually map single cells into their X and Y coordinates. And then you also retain those kind of cytosolic transcripts. And so most of the stuff that I'll talk about today is using the Visium platform from 10x Genomics. And I'll talk a little bit about some data that we're generating from other platforms. But basically, just briefly how this works is that a tissue section is cut, it's put onto a slide, and these slides have basically spatial barcodes. And so there's little spots on there. When you do the cDNA synthesis on the slide, spatial barcodes get incorporated. You take an image of the tissue, and then you basically are doing sort of an RNA sequencing experiment on each of those little, kind of regions of interest or spots. So in this case, in the Visium platform, there's 5,000 of those spots. You're kind of doing 5,000 RNA sequencing experiments across the tissue. And then you can map those individual experiments back to the exact X and Y coordinates. So you can kind of build that back in X and Y 2D space. I will note that the resolution of the original platform, which I'll talk about, is not awesome. And so we're really working with 50 micron spots. And in the human cortex, that works out to be something on average about 3 cells that will be, or 3 cell bodies that will be in there, although it varies. But we can segment the images, so we do know how many cells are in there. This is rapidly advancing technology. So now the technologies that we're using now and generating data with currently are at single cell resolution. So then the part that I talked about before is, so what do we do with this data? Like, why do we want it besides just generating data for data's sake? And how do we get to the molecular associations of disease? And so really, we sort of take 2 approaches here to map disease associations to individual single cells from the single nucleus RNA sequencing, and then also to spatial context. So approaches that we use are to map genetic risk. So we do things like kind of curate GWAS association data, so genetic risk, also from TWAS, so it's from large-scale transcriptome-wide association studies, and then also looking at across these disorders that we care about, highly penetrant or rare variants that we know of that contribute to disease. And so we can basically take those lists of kind of genetic variation that contribute to disease, and then contextualize that data to understand like how might those genetic variations be contributing to disease. We can project that into the different cell types that are in different brain regions that we're sequencing from to understand where is that genetic risk enriched. So is it in a specific cell types or across cell types, and then where in the brain? And then we can increasingly do this with the spatial expression and integrate these 2 together so that we can really tell you where genetic risk is, which cell types, and then where in the brain. So this is sort of, I'll go into this a little bit later, spatial transcriptomics data in the cortex where we can look across the histological layers. And so we can really kind of understand where that genetic risk is lying in terms of within the intact tissue structure. The other approach that we really take is to take curated gene lists from animal studies, from model organisms, and also from the 20 years of large-scale bulk RNA sequencing studies that have been done in case control cohorts across really large hundreds of populations. So those studies didn't have any, cellular resolution, and they didn't have spatial resolution, but we can really capitalize on all that data that had been collected over the past 20 years, go back and take those differentially expressed genes from these large scale case control studies, and then really kind of contextualize them to give them cell type specific information and spatial information by projecting them into those molecular profiles for the cell types and into the molecular profiles for the spatial domains. Okay, so I'm going to basically now go through sort of 3 examples of studies that we've recently completed to sort of illustrate how we do this and to find these molecular associations. So the first, I'm going to talk about how we map spatial enrichment of kind of biology or function and disease signals in the human dorsolateral prefrontal cortex. Second, I'll talk about work that we're doing to identify spatial gene expression in the human hippocampus that's relevant for disease, but then also for circuit connectivity, which is important to us since we know that circuits are important for these disorders. And then last, I'm going to talk about some spatial molecular signatures of neurogenesis in the human dentate gyrus. And I put this one in here not because it's directly sort of linked to disease, but because I know there's a lot of people here who are very interested in hippocampal neurogenesis. So I felt like I would do this special one for this audience. Okay, so basically, why care also, about spatial, besides, the things that I talked about, that we get the cytosolic transcripts and just that you want, the spatial information. And when we started doing these studies, we did them in the dorsolateral prefrontal cortex. And part of the reason for that is we know that that region of the brain is very involved in a lot of the disorders that we care about, schizophrenia, mood disorders. But also, we really thought if we're going to go into starting to do spatial transcriptomics in the human brain, what's a region that sort of like exemplifies, why you want structure, function, and why you'd want to see gene expression within the spatial context. And the DLPFC or any cortical region is really beautifully kind of, illustrates that. So even if you look at these really old, like histological stains here, you can appreciate that, like, if you were just going to do single cell sequencing and you smush this tissue up, like you wouldn't know where the cells came from. And just looking at this, you can appreciate that, right? Like where they come from, they look different. So it stands to reason that the gene expression kind of, or the molecular profiles that are dictating where they are and what they look like within that space is going to be important. And also as neuroscientists, we know this to be true, right? So there's decades of research on the cortical layers that suggests that where they are matters for a lot of things, particularly for the wiring. So across the layers, where neurons come in and where they go out and who they connect to is dictated by where they sit in which cortical layer. And we know a lot about this. And then also across the cortical layers, you can really kind of appreciate that here. These cells really look different, right? And so we know that the physiology, the morphology of these cells is different across where they sit. And any of that structure function is going to kind of back up 2 steps to be that the molecular, something's driving that, right? And it's the gene expression. And so that's why we really wanted to, when we first started doing this, we really wanted to kind of look at this spatial gene expression within the cortex. So when we first did this, I think it's like going on 4 or 5 years ago now when we decided to try and create the first spatial molecular maps in the human brain. We were using the original Visium platform. And so the way that we did this is from our original study. We cut these tissue blocks in the DLPFC, basically from the peel surface, coming down through, retaining all of the layers into the white matter. And then this is what, this is a piece of tissue that gets put on a cryostat chuck. We kind of cut a 10 micron section. And then this is a picture of the tissue section just laid down on one of those spatial transcriptomics arrays and then stained. So we do a histological stain, it's an H&E stain. And you can appreciate here just by seeing the morphology that you're starting with layer 1 at the peel surface down through layer 6. And you can see, the white matter here. When we did the Visium platform, this is transcriptome-wide, so it's basically across the whole genome. This is just pulling out 2 genes, and this is just a gut check to show you that, or convince you that this works, right? So we picked out SNAP25, that's a neuronal gene. We picked out MOBP, which is a gene that maps, kind of represents white matter. It's expressed in oligodendrocytes. So the warmer colors here are higher log counts, and so that, more gene expression. So you can see SNAP25 is in the gray matter, in the layers, that makes sense. And MOBP is really enriched in the white matter. So this felt really good. It kind of platform worked. At this point, with the first study, what we really wanted to do was just, because it hadn't been done before in the human brain, was make molecular profiles for the individual layers. At the time when we did this, there were no data tools to analyze this data. So there was no way to cluster the data in an unsupervised way. That's changed a lot, I'll talk about. So at this time, we actually manually annotated all 5,000 of these spots. This was like one of the worst experiences of my life. But we basically did this thinking, we know this. I'm looking at the histological image and then also marker genes. So we went through this like in this whole study and basically manually annotated all of these to create sort of a ground truth or a reference for these. And then once we did that, what we could basically do is pseudobulk, which just means that we took those spots that we had annotated as layer 1, 2, 3, 4, 5, or 6, or the white matter, and then kind of like mix, mush them together and then sort of collapse them into spot level data and collapse them into this layer level data. And by doing this, we just basically made the first molecular profiles for individual, layers in the cortex. And by doing this, we found hundreds to thousands of differentially expressed genes to generate these molecular profiles, and compared this to a lot of the rodent data, and surprisingly found that a lot of it was not conserved, which some of it was, but some of it's not. And I think that just points out that these studies are important to do in the human brain, because while mouse models or rodent models generally are really important and useful, they also have differences in the human brain. We've gone on since then, so this technology has been evolving really fast. This was our most recent study, so we did a much larger scale study in the dorsolateral prefrontal cortex, and I'll just kind of run through some of the highlights from this, like as we've expanded the technology, what we've been able to do with this. So in this study, we basically did a within subject comparison over the anterior to posterior axis to get asked a lot about this. We're taking just a little 10 micron section. Do you have any kind of changes, not just in 2D space, but in 3D space? We also did some spot level deconvolution to integrate protein data. And then we identified ligand receptor pairs using sort of cell-cell communication across the spatial domains to find some ligand receptor pairs that were related to schizophrenia risk. I'll talk about in a second, I'll show 2 slides on this, that we also, in this case, were able to use unsupervised clustering to identify novel spatial domains beyond the actual layers, and then use the data to do spatial registration to integrate diagnosis-related data. So here I talked about how with our first study, we manually annotated all those 5,000 spots because there were no tools to actually do unsupervised clustering. So in the 4 years between when we did that original study and then when we were doing this, we also had developed a lot of kind of unsupervised clustering tools to actually work with this data. And this was really awesome to be able to do. So this is basically showing you, this is not a manual annotator, this is a computer doing this. So what you can do is do clustering, spatial clustering of the data. And here we're asking, for example, give me 9 clusters, give me 16 clusters, or give me 28 clusters. And so when we basically clustered at 9, we could really well represent the 6 histological layers in white matter, and then actually break them out into kind of sublayers, which we know from the literature do exist. And then basically going up to higher K, what we started to find was that we could find spatial domains that we couldn't see with our eye. And this was really exciting because, it's kind of cool to get the molecular profiles for the layers, but also we know they exist. And kind of the power of this technology, what you want it for is also to find stuff that you can't see with your eye. And so some of those things that we actually found, which we're really excited about, is that we could find spatial domains for that were associated with vascular and also with immune function. And so this is really interesting for us, I think, because there's a lot of information coming online that a lot of these disorders, that there's kind of issues with the neurovascular niche and also immune signaling. So we can look for enrichment of genetic risk across these, not restricted to just the canonical histological layers. So another thing that we can do with this is basically spatially register disease signals. So this data was actually part of a really large kind of data generation across the PsycEncode consortium. So a lot of the other studies on that PsycEncode 2 collection had been generating a lot of case control data. So we basically, but it was single cell data. So there was massive millions of nuclei across different disorders. And so what we did was basically spatially contextualize those case control findings. And so this is an example of this that we did from some of that data, where we basically took case control single cell data from neurotypical controls versus individuals that had been diagnosed with autism. And then we're able to take those differentially expressed signals across the individual cell types, which are basically down here on the x-axis, and then actually spatially contextualize them across our spatial domains. So that we did this for the autism study, We also did this for PTSD and major depressive disorder. And we're able to really, with the MDD and PTSD, which was really interesting to us, really localize those kind of differentially expressed and genetic risk signals to layer 1 into the meninges. We're mapping onto a lot of these kind of immune and vascular systems. Okay, I'm going to move on to talk about some of the work that we've done in the human hippocampus. And so this study was a more recent study where we really set out to do a more difficult region than the dorsolateral prefrontal cortex. Many of you are familiar with the hippocampus, really important for learning, memory, and a lot of important functions in neuroscience and implicated in a lot of the disorders we care about. This study was very hard to do because the human hippocampus is kind of big. So when we were doing the original studies in the DLPFC, we could cut a cortical column that encompassed, all of those layers and put it just on one of these arrays. The hippocampus is really big, so we could not do that. And so here, basically, we had to, dissect these pretty large blocks, score the tissue, and it took us basically 4 capture areas or 4 of those assays that we had to sort of mount them on, stitch them back together, and then computationally integrate the data to create a composite of the hippocampus. So this study here, we basically did 10 neurotypical donors. Also, this is for those of you who are interested in this or want to use these data as resources. These are the same 10 donors that we did the other study that I just talked about from the DLPFC. This opens a lot of opportunities because we have the same donors and have both regions. And for us and many other people are interested in hippocampal prefrontal circuits. And so we can really look at gene expression across the circuit there. And so here we did paired Visium and single nucleus RNA sequencing from those 10 donors. So we're cutting the sections for Visium and then also collecting, adjacent sections that we're doing single nucleus RNA sequencing on so that we can integrate those data to really get cellular resolution with the spatial resolution. So this is what this data looks like. So again, on a slide, we needed 4 of these capture areas. So this is a section through the hippocampus and how these kind of overlapping sections were placed. We do the Visium assay and then get the transcriptome-wide profiles. This is an excitatory marker. You can kind of appreciate, again, like warmer colors. You're seeing it mapped to kind of the, cell body layers. Here, again, we went back originally and did the histological manual annotations because this was a long time ago when we did this, so I didn't have to do this, somebody else did it. But we did manually annotate all of these kind of canonical hippocampal domains. And so we were able to see from kind of, gene expression markers and then also the H&E images to look at sort of all the known hippocampal domains we were able to annotate. We did take this data into the unsupervised clustering. And again, this is something that's really was, kind of exciting for us, is that the unsupervised clustering was really able to identify a lot of features that were missed by the human eye. So these are the manual annotations, and you can appreciate sort of the dentate, CA3, CA1, and sort of the main things. With basically the unsupervised clustering, we were able to kind of pull out a lot of different sort of inhibitory and finer grained mapping. So this is really useful for us that we can kind of find these spatial domains that we can't see with just the manual annotations of the human eye. So what do we do with this data once we had these unsupervised clusters across the hippocampus and the single nucleus RNA sequencing data? Again, this is one of these approaches where we are taking GWAS data to basically ask where is sort of genetic risk across these neuropsychiatric disorders enriched in the hippocampus. And so we did this for the spatial domains and for the single cell data. This is kind of depends on what you're interested in here. We found some really interesting things, basically, where we find enrichment for schizophrenia in some of the CA1 domains, and then that also kind of maps with the single cell data. We found a lot of enrichment across the retrohippocampal formation, which was sort of interesting to us. We're not expecting that. And so some of these things, are basically things that we're going to go back and follow up to kind of try and contextualize what those actual genes are and what's interesting about them. So here, I think the next thing I'm going to talk about is that those kind of approaches are sort of simplistic of basically just saying like, hey, here's the molecular profile for a spatial domain that either we annotated or that came out of a program to unsupervised cluster, and then sort of looking at enrichment of genetic risk or some kind of gene list. What we really wanted to kind of find also was to use more sort of sophisticated computational tools to find patterns in the data, to find sort of more fine-grained spatial things across, so not just within one spatial domain, but across the spatial domains. And the thing that we'd use this for, approach we used was non-negative matrix factorization, which is basically a machine learning approach to find latent factors in the data. So basically what this is going to do is just kind of find patterns in the data. Because there's some limitations. This is just here kind of like spatial registration where we're mapping individual single cells to their spatial, and it misses a lot of things. Like I said, it can't kind of go across different spatial domains. And so the way that this works, I'm not going to go through all the math, it's a little bit complicated, but basically we're doing this with the single cell data. So we're just looking for patterns in there. And then what we're going to do is take those labels that we find and then transfer those patterns into the spatial data. So if we find these patterns across the single nucleus RNA sequencing data that can go across different spatial domains or across different cell types, we can then project them into the spatial data and look for those patterns. And I'll show you a couple of examples of this. So here, one of the things when we did the unsupervised clustering that we realized, and part of the reason that we wanted to go after these more kind of fine-grained patterns, is that we couldn't actually kind of resolve between CA2 through 4. So when you look at this unsupervised clustering map, we could really resolve CA1 fine, so that's in this green color. But the molecular profiles for CA2 through 4 are so similar, like in the gene expression, there's so much overlapping that we really couldn't resolve them. But we know that they're different, right? Like, you know that CA2, 3, and 4 exist. And so this is really part of the reason, this is one of the reasons that we wanted to go after these more sophisticated approaches. And kind of long story short is this is an example where this did work really well. And so by doing this, we basically, if you go back here, when we did this NMF pattern generation, we found 100 different patterns. And a lot of those patterns were just kind of like cell type specific. And some of them were just for a spatial domain. And the ones that we're really interested in might be individuals that, patterns that go across different cell types and the spatial domains. And here, what was really exciting for us is that we were able to find patterns that really could differentiate between mossy cells, CA3 neurons, CA2 neurons, and CA1 neurons, and then find those areas in the tissue, which we could not do with unsupervised clustering. So this is just an example here, this green one of a pattern. The numbers are arbitrary, which basically, is mapping to mossy cells. And you can see this when we project it onto the spatial data, that it's actually able to find that, which we couldn't do in the unsupervised clustering. And same that we can do that, for the CA3 neurons, for the CA1 neurons, and for CA2 neurons. The CA2 was really exciting because people have had a really hard time. It's really small to actually find unsupervised clusters that map CA2. So this was really nice for us that we could do this and kind of really map this very cleanly that you see in this blue color here. Another thing that we wanted this for is to basically kind of do spatial mapping and inference of circuit connectivity. And so where we're really interested in doing this was sort of in the CA1 and subiculum, so the output regions. So those regions, right, are important because, we care about the hippocampus, but really the important thing also is that the information gets out of the hippocampus. So those CA1 and subiculum neurons are going to project to areas that we really care about and transfer information. And we know that a lot of those circuits are involved in these disorders. But like I talked about, we can't actually look at circuit connectivity in human brains. We can't label projection neurons or actually do molecular profiling across the circuit. And so our approach here was to think about how we could capitalize on this, rodent data where you can do those approaches. And so the first thing that we did was really look across, so focus in on CA1 and those subicular regions in the data to see if we could find individual patterns that might look like they have spatial kind of patterning that looks like it would be, could map onto different areas of projections. And we were able to find that. So within, we find patterns in CA1 versus different areas of the subiculum and the presubiculum that we can map out. And what we were curious about is those individual patterns, may they actually be kind of mapping onto a population of neurons that is where the spatial topography is like that because they project to a specific region. And so again, there's no way to do this in the human brain. We can't actually go and put tracers in there and know. So what we wanted to do here was to basically capitalize on rodent data. And so a large kind of study had just come out last year from the Brain Initiative BICAN studies, where they did these kind of mapping of projection neurons across the whole brain and then sequenced from them. So we went and grabbed that data. Basically, that molecular data, has these molecular profiles, including from the hippocampus, from the output regions to different areas. So basically here, what this is on the y-axis is different regions, right? So these are the molecular profiles from a mouse that are projecting out of the hippocampus to these different projection regions. So the PFC, the amygdala, the thalamus, the striatum, piriform cortex. And then what we did was take our NMF patterns from the output regions, so from CA1, subiculum, or from the retrohippocampal formations from layer 5-6 or layer 2-3, where we had found these really kind of restricted space, where the populations, these patterns had really kind of defined spatial topography, and then asked, do these kind of enrich or map to these, projection neuron profiles? It's not perfectly clean, but like the kind of really neat thing about this is that this approach does work. And I think right now, this data is somewhat limited. The mouse data is quite sparse, but we can find individual patterns that really seem to allow us to infer that that's a population of neurons in the human hippocampus, in the subiculum or in the CA1 regions, that we can infer the projection targets of those. And I think that's really important because that allows us to really capitalize on these studies that we can do with circuit, manipulations and things like that in rodents to know that those neurons in the human brains are ones that are going to go to PFC versus striatum versus thalamus, for example. Okay, and last, I'm going to talk about for the last bit of the talk about how we have kind of used this data to look for molecular signatures of neurogenesis in the human dentate gyrus. And again, I think, part of the reason for us doing this was just interest, that using the spatial data would be really useful because hippocampal neurogenesis is localized in the granule cell layer of the dentate gyrus. So, people who have done single nucleus RNA sequencing studies, they're looking for those, new neurons, but it's smushed up tissue, so you can't really kind of put them on the map. And we know that those, that there is a neurogenic niche. So using the spatial technologies was really attractive to us to be able to look for this. And also there's a lot of interest in hippocampal neurogenesis as a treatment, and also associated with a lot of disorders like depression and the response to antidepressants. So there's a huge literature around this in the rodents, but there's been an ongoing controversy about whether this exists in the human brain or to what extent it exists in the human brain. So we also really wanted to capitalize on the ability to use the spatial data to be able to localize those neurogenesis signatures. I just want to give a shout out here to Tony Ramnath, who's now a postdoc in Mora's lab, who this was his graduate student thesis in my lab. And so he did a really excellent job on this project. It was actually just finally accepted at Cell Reports, and so should be online or in press soon. And basically what he did for this study is we had 4 donors for each kind of cohort, for each group across this cohort. So we took infants. So these were donors that were brain donors that were between 2 months of age and about 18 months of age. We had a teenage group, an adult group. These are just sort of middle-aged donors, and then elderly donors. And here, instead of profiling across the whole hippocampus, because one of our kind of questions of interest here was really about neurogenesis, was to focus specifically on the dentate gyrus. So here we just did one Visium array instead of this approach of kind of, stitching them together across the whole hippocampus. And so here we're going to look across the lifespan to look at changes in the dentate gyrus, specifically kind of trying to see if we can find molecular signatures of neurogenesis. And so what Tony did here is basically the same as what I talked about before. So he generated this data and then used unsupervised clustering to be able to kind of really fine sort of resolve spatial domains within the dentate gyrus. This worked really well. We could, in addition to just saying like, hey, this is spatial domain for the dentate gyrus, we were able to kind of subdivide or subcluster out within the dentate gyrus. So we're able to really well resolve, for example, the granule cell layer, so important to us, thinking about neurogenesis, as well as the subgranular zone molecular layer. And then on these arrays, some of them we're focusing on the dentate gyrus, but some of the donors we are picking up parts of CA3, for example, but not kind of consistently in all the donors. So for this, we were able to identify domain-specific markers for all of these different kind of subclusters within the dentate gyrus, which is nice to have and to be able to compare them to what's been identified in the rodent. And so once we had these, we basically did this approach of pseudo bulking those spots that I had talked about before. So that basically means that we're just kind of taking like any of these colors here. We basically take those spots and kind of grab them all together, and then are going to look at gene expression across the individual spatial domains. And then here, we're going to do that across those clusters, so across the individual spatial domains, but then across the age cohort, so across infants, the teens, the adults, and the elderly. And so this is just a principal component plot. And basically really fast when we did this, what we saw here is that the main variation in the data is spatial domain, which is not surprising. So basically you can see across the first principal component, if you look here, which are the spatial domains, so the colors, you can see that most of the variation in the data is really by spatial domain. So these domains have quite different gene expressions, which is not surprising. But in the second kind of component of variation, or the thing that drove the second most was age. And that really was driven by the infants. And so if we looked at this, it's really, you can kind of see this here by drawing these two circles, that down here on the second principal component, these squares are the infant. So that variation in age is pretty much purely driven by infancy. Second by elderly, but is really driven by infancy, suggesting that the dentate gyrus, that the molecular profile of it in infancy is very different than the rest of the brain or the rest of the age, which is not surprising to us. When we basically kind of just take these across infant, teen, adult, and elderly across these different domains and do sort of gene ontology to look at sort of what signaling pathways are enriched across those, we find that there's sort of opposing processes of proliferation and neurogenesis. So these are kind of hard to read here, but these are the infants. So the blue is downregulated kind of signaling terms, and the red is upregulated. And so basically what we see is this kind of opposing. If you look at the infants versus the elderly, it's kind of opposite. You see that sort of there's a response to ischemia, like things associated with sort of that we associate with senescence and inflammation are downregulated in the infant and then upregulated in the elderly. And then these terms that are kind of clustered down here are related to neurogenesis. And you can see that these are very strongly represented in the dentate gyrus of the infant, and then are not very well represented anywhere else across teens, adults, and elderly. So here, once we knew this, that kind of the infants were sort of driving this variation, we basically did an analysis here where we just looked at infants versus non-infants. So we collapsed the teens, the middle age, and the elderly, and then just basically looked at gene expression in specific layers. So here I'm showing you in the specific spatial domains, just the granule cell layer at infants versus non. And here what we found is lots of canonical neurogenesis markers, so double cortin, and then also was one of the top genes differentially expressed, not very surprising. And then a lot of proliferation markers, and then also a lot of extracellular matrix components that have been associated with neurogenesis. Next, what we wanted to basically do is really capitalize on sort of this ability that we had these spatial domains and could isolate the granule cell layer in the subgranular zone. So we took a lot of existing data that was in the literature of molecular profiles for kind of different profiles for neurogenesis across the maturation trajectory. So starting from sort of proliferating cells, neuroblasts, migrating neuroblasts, anything that we could find in the literature where we had sort of single nucleus RNA-seq profiles. We did this from mouse, from non-human primates, and also from humans. And basically the take-home message here in this heat map is sort of, we're looking at the infants, the teens, the adults, and elderly. And then we're mapping those molecular profiles that we're pulling here for different kinds of stages across neurogenesis, and then projecting that into our spatial domains to see if they exist. So we basically do find that across the lifespan, we do find evidence of these different across the lifespan. So you can see here, this is the granule cell layer. We find enrichment, and it's highest in the infant, which makes sense. But we do find it in the teens, the adults, and in the elderly at the granule cell layer. So those kind of markers or molecular profiles that have been pulled, and defined from non-human primates and from mouse, they do kind of map into the granule cell layer. But the thing that was kind of less satisfying to us is that it's not just the granule cell layer. So you can see that we also get a lot of sort of, mapping of those neurogenesis signals into other regions. So into the CA regions and things like where they shouldn't be, I guess. And so this felt to us kind of unsatisfying. That was not very clean to know, like, yes, these neurogenesis genes are across the lifespan in the infants, they are at the GCL, but not just at the GCL. And so what we wanted to do then was, because this is sort of messy, just this sort of like enrichment thing across spatial clusters, like I talked about, is go back to this sort of more sophisticated computational method of looking for more specific patterns in the data. So we went back to this NMF kind of pattern finding, approach that we had used in this earlier study, and thought that maybe here we could kind of find more specific patterns for dentate granule cells. So again, basically what we're using here is this same, the main hippocampus data that we had generated earlier, and taking those NMF patterns that we identified with RNA-seq and then projecting them into the spatial data. So this is just a UMAP plot of the single nucleus RNA-seq data that we had generated in this really large scale, hippocampus project. And over here are subclusters of granule cells. And so from those 100 patterns that we had originally generated, we kind of projected them into the data to say, can we find patterns that are specific for granule cells? And the answer is yes, and there's 3 of them. So they're in this kind of magenta color, the yellow, and then this green. And you can see here that they're sort of in a non-overlapping population. And so what we thought is that those individual NMF patterns that we find may be specific for different types of granule cells. And this was interesting to us because we wondered if they might represent sort of a maturation trajectory. Maybe there's some young ones, some migrating, and some old ones. And so once we had found these, we basically then projected that into the data. And interestingly, there is a spatial kind of component to that. So this sort of one that's in the magenta here, you see is kind of deeper. And then the other patterns seem to be more superficial if you're looking at this across the granule cell layer. So this was interesting to us, suggesting that this kind of wondered to us, like, are these potentially younger cells? And it seemed like that. So when we basically did kind of just gene ontology and looked at what was like represented in those patterns, we found that this kind of magenta marker here, that the genes that are driving that pattern are actually kind of neurogenesis early progenitor genes. And then things that were kind of represented in this yellow and the green pattern were things that are associated with more mature dentate granule cells. And then in this green one here, that's the most superficial, we find some markers that are associated with senescence and inflammation. So this kind of said to us, we wondered, are we actually mapping here sort of a trajectory of early, migrating sort of or proliferating granule cells, more mature, and then aging granule cells. And so we basically then use this kind of projection technique, or this label transfer, to project this into the spatial data across the lifespan. And so when we did this is the case. It's kind of really exciting that we can basically find evidence for the existence of a maturation trajectory for these dentate granule cells across the lifespan. So that purple or magenta pattern that we found that had kind of had these markers of more early cells or proliferating cells, when we project that into the spatial data across the lifespan and look specifically at that spatial cluster for the granule cell layer, you can see that it's only in the infant samples. So it's not at all in the teens, the adults, or the elderly. And then we find this kind of other pattern where it's sort of more in the middle of the granule cell layer and has markers of more mature. We find that this really peaks during kind of adulthood. And then this third pattern here that we found, when we map this into the spatial data, we really only find this in the elderly donors at the granule cell layer and is absent from the infants, the teens, and the adults, suggesting that this is kind of a real pattern or specific markers for maybe granule cells that are approaching senescence. And so now we basically are kind of taking that data, and I'll talk just a little bit now about what we're doing with all this data, because I get asked this a lot, like, you guys generate all this data, but then do you do anything with it? And so we have sort of an ongoing focus on those molecular signatures for immature granule cells, which we're really interested in. So again, this was sort of this gene expression where we were looking at just the granule cell layer in the infants versus non. And some of these genes are these canonical, neurogenesis markers. But we are really interested in some of these extracellular matrix components. For example, this gene here, posted, which encodes the gene, the protein periostin, which has in rodent models been associated with neurogenesis. It is also a marker here, as you can see, for this NMF pattern that marks these immature granule cells. So also, as I talked about the resolution of that Visium platform, which is what we did the study with, we don't really know. I mean, it seems like those are marking new neurons, but we don't have cellular resolution because the spots there or the individual ROIs, there's about 3 to 5 spots. And actually in the granule cell layer, it's more than that because the cells are so packed. And so here we really wanted to validate some of these findings with these immature sort of markers and different sort of markers of the granule cells with something that has true cellular resolution. So this is data that we took back in using the Xenium platform from 10x Genomics. So this is an imaging-based platform for spatial, where we basically can look at cellular, or it's actually subcellular resolution. And so this is also just a really beautiful picture of one of these. And so we're basically here, this is targeted, so it's not transcriptome-wide. So there's 5,000 genes on this panel, and we can dictate some of those genes to put our genes of interest. And then some of them are panels that are sort of kind of collated around different sort of interest, neuroscience or whatever. And so some of these genes that we're interested in, we have on this panel. So just to kind of show you what this looks like, this is this gene periostin, which is an extracellular matrix factor that we find to really be a marker of this pattern for immature granule cells. And this is from one of the infants. And so you can see here, we can now show that that gene is really marking these immature granule cells because this is at true subcellular resolution and is co-expressed with a lot of kind of canonical markers of neurogenesis. And just to kind of show strikingly how much this declines over time, if you look just from like in one of these teen donors, you can see how rapidly this decreases and changes to, we're really kind of finding very low levels of this. And at this point now we're kind of following up on a lot of these things to take these back into, induced pluripotent stem cell models that are hippocampal-like. So we can take these back in and kind of modulate these particular genes. And this is really of interest to us, some of these, markers that we're finding of these immature cells in the infant that we can try and overexpress or manipulate to kind of increase cellular rejuvenation and things like that. So we are doing those in these iPSC-based hippocampal models at the moment. And so I will finish up because I'm at time and give you a summary. So I talked to you today about the spatial transcript, our early work doing spatial transcriptomics, generating some of the first kind of transcriptome-wide maps in the dorsolateral prefrontal cortex, and how we use that to map spatial enrichment of genes and cell types that are associated with neuropsychiatric disorders. And then I also talked about sort of capstone project that we did to generate sort of the first transcriptome-wide profiles across the human hippocampus at cellular and spatial resolution, and sort of how we can use those to integrate with rodent models of circuit connectivity to infer projection targets out of the hippocampus. And then finally talked about sort of this focus study in the hippocampus on the dentate gyrus to specifically focus on changes across the lifespan. There's lots of stuff in that paper also about the other end of the study about aging, which I didn't have time to talk about, so hopefully you can check that out. And with that, I just will give my acknowledgements. I talked already about Kristen, Stephanie Page, Leo Claudio Torres, and Stephanie Hicks. And then I think across the talk, I had a number of people who've collected the data. Anthony, as I said, did all of the kind of last study on the dentate gyrus. And then Jackie Thompson, Eric Nelson, and Maddie Timpani did sort of all of the work on the main hippocampus project. And I'd be happy to take some questions.